Hello students, welcome to our online video tutorial class. I think you have seen the previous video whatever I have made based on the introductory part of the chapter reproductive health, as well as I have also discussed there regarding the various birth control methods. So in that video I have mentioned at last that I will be starting from the amniocentesis portion in the next video. So today I have come with the next video starting from the next portion, that is the amniocentesis. We all know amniocentesis is generally a method by which the amniotic fluid is collected from the amnion, and after collection of this amniotic fluid, obviously that will be analyzed for determining the genetic disorders present within the fetal skin cell, as well as not only the genetic disorder, with that various kinds of metabolic disorder, sex of the baby. These all things can be determined from the type of cells, specifically the fetal skin cells present within the amniotic fluid. So that analyzing method is commonly known as amniocentesis. First of all, when we are talking about the amniocentesis, we need to know regarding the purpose of the amniocentesis, why we will perform amniocentesis. First purpose of the amniocentesis is to determine the genetic disorder, if any kind of genetic disorder, as an example Down syndrome, Klinefelter syndrome, Patau syndrome, Edwards syndrome, any kind of genetic disorder is present in the newly born baby or not. For determining that, obviously this specific amniocentesis process is so much important. Second thing is that determination of metabolic diseases, that means any kind of metabolic diseases, as an example alkaptonuria, phenylketonuria, this kind of metabolic diseases that are also present in the newborn baby or not, that can be also checked by this specific procedure. Third point is to study regarding the sex of the child, whether the individual is male or female who has taken birth. That can be also finalized, or that can be also determined by this specific type of process, that is amniocentesis. Next one, to perform the chromosomal analysis, that means whatever the chromosomes are present, their structural analysis can be done by this specific process of amniocentesis. And at the last, we can say regarding the karyotyping. Karyotyping means the chromosomes which are present in the newborn baby, arrangement of the chromosomes according to their number, according to their sequence. So that specific sequencing of the newborn baby's chromosome, and with that the structural analysis of the chromosomes, that can be also done by this specific process of karyotyping, and as well as by this specific type of the process, that is amniocentesis. So for these 5 different purposes, amniocentesis process is very much important to perform. Coming to the next point, that is the mechanism, that means how we can perform the amniocentesis mechanism. So for that, first of all, we need to collect the amniotic fluid. A very little bit amount of amniotic fluid has to be collected from mother's womb. After collection, we need to perform the centrifugation, because the type of cells which are present within that specific amniotic fluid is a fetal skin cells. So we need to precipitate those fetal skin cells. Otherwise, if we will not precipitate those fetal skin cells, obviously we can't carry forward our experiment, or we can't carry forward our process of analysis. So for that reason, amniotic fluid has to be rotated by the centrifugation process, and after centrifugation, obviously there should be 2 parts, one is pellet, another one is supernatant. So in the pellet portion specifically, we will be obtaining the fetal skin cells. After obtaining those fetal skin cells, now we can go for various kinds of analysis, any kind of genetic disorder is present or not, any kind of metabolic disorder is present or not, what is the sex of the baby, whatever the type of karyotyping process, those all work, structural analysis of the chromosomes, these all kind of activities we can do by that specific or by those type of fetal skin cells. So for this purpose, actually the amniocentesis we are commonly doing for analyzing these all things, and this is the mechanism of amniocentesis. Now we are coming to the next topic, and that is a fully different topic than from the amniocentesis, that is medical termination of pregnancy. What is this? Medical termination of pregnancy is a such kind of the process where before the full development of the newborn fetus within the mother's womb, forcefully the growing embryo or the growing zygote it will be getting destroyed, and after destruction it will be taken out from the mother's womb. So it is entirely, it is an induced process, it is not a natural process. Human interference is obviously needed. So this kind of process where in a forceful manner the growing embryo or the growing fetus within the mother's womb, from there we will be withdraw it, that specific process is commonly known as medical termination of pregnancy or induced abortion. So it is written here, it is an intentional process or voluntary termination of pregnancy before full term is called, before full term means before further development or full development, and that is called as medical termination of pregnancy. Keep it in your mind, if medical termination of the pregnancy has to be performed for any of the female, we need to keep it in our mind, we need this medical termination of the pregnancy need to be done during the first trimester, that is up to 12 weeks of the pregnancy. Up to 12 weeks it should be done. Second trimester abortion is much more riskier, because after 12 weeks, that means after first trimester, if this MTP or the medical termination of pregnancy will be done for any of the female, it will be riskier for that mother as well as riskier for the child. So obviously we are going for the abortion where the fetus is actually getting destroyed. So fetus is will be destroyed, it is a normal thing, but the mother life also can become riskier if the MTP will be performed after 12 weeks or after first trimester. Now we need to know why we will perform the MTP or medical termination of pregnancy or induced abortion. These are the following reasons for which purposes we will be performing MTP. First one, women whose physical or mental health were endangered by the pregnancy. Okay, so in case of those of the female whose physical health or mental health is getting spoiled or getting endangered by due to pregnancy, obviously for those kind of female, MTP need to be performed. Women facing the birth of a potentially handicapped or malformed child, if it is analyzed properly if it is determined beforehand that the newborn baby is handicapped or the newborn baby is genetically disabled, if the newborn baby they have some genetic disorder, so obviously if the female or if the mother wants to abort, she can do by this process of the medical termination of pregnancy. If the newborn baby is the product of any rape case, so obviously if the mother wants, then on that case by the process of MTP, that specific newborn baby or the new growing baby that can be killed or forcefully withdrawn from the mother's womb. Pregnancies in case of unmarried girls under the age of 18 with the consent of a guardian. So under the consent of the guardian, if any female is getting pregnant below the marriage and below the age of 18, so obviously by taking the consent of the guardian, MTP can be performed. Pregnancies that are a result of failure in sterilization. So if any kind of pregnancy can be created and that is a result of any kind of failure in sterilization, that can be, on that case also, this MTP or induced abortion has to be performed or can be performed. So now these are the reasons for which the MTP or medical termination of pregnancy we are commonly performing. Now we are coming to the next point, that is the mechanism. What is this? Mechanism is how we will be performing the MTP or medical termination of pregnancy. So there are actually three mechanisms. First one, by prostaglandin injection. Prostaglandin injection is very much important for the medical termination of the pregnancy, because prostaglandin, when we are injecting, automatically what is happening, the contraction and relaxation of the uterine muscle, it is getting started, and when the contraction and relaxation of the uterine muscle is getting started, automatically it will be easier during the time of MTP or during the time of induced abortion to withdraw the growing embryo from the mother's womb. Vacuum aspiration, this is the method where we were using a vacuum cup by which, or you can say rather vacuum aspirator, by which we will be withdrawn the crushed embryo which is present within the mother's womb. So first of all we will be crushing the embryo within the mother's womb, that means we will be killing that. After killing, by using the vacuum aspirator, we will be just withdrawing it out from the mother's womb. Third one is dilation of cervix, that means cervix dilation is also very much important. For that we will be applying relaxin, because if the cervix will be getting dilated properly, on that time obviously it will be easier to perform the medical termination of pregnancy or induced abortion. So these are the three methods by which induced abortion or medical termination of pregnancy can be done. Now we are coming to the next point, and that is a very important point, that is the STD, sexually transmitted diseases or sexually transmitted infections. So let's see whatever we need to study under this. First of all, the definition of STD. What is the STD? STD is such kind of disorders which are getting transmitted from sexually unhealthy person to the sexually healthy person during the time of unprotected sexual intercourse. So if during the time of unprotected sexual intercourse, the various disease causing agent or even the disease only is getting transmitted from the unhealthy individual to the healthy individual, that transmission is commonly known as sexually transmitted diseases or reproductive tract infection also. So it can be also called as RTI or STI, that is a sexually transmitted infection or reproductive tract infection. Now the point is, whatever the types of sexually transmitted disease are commonly happens in case of human being, there are several kinds like that: gonorrhea, chlamydiasis, syphilis. These three are actually the bacterial sexually transmitted diseases because these are caused by the bacterial agent, as well as genital herpes, genital warts, trichomoniasis, hepatitis B, HIV/AIDS. These are the viral STDs because these are all caused by the viral agent or virus. So these are the types of STD which are commonly happening in case of human being. Now the point is mode of transmission, that means how these STDs are getting transmitted. First of all, obviously the diseases can be transmitted by the help of bacteria, and those kind of diseases are chlamydiasis, syphilis, gonorrhea, already I have told. These kind of diseases can get transmitted by the help of viruses also, as an example AIDS, hepatitis, genital warts, genital herpes. These are the viruses are also present for transmitting the diseases. This specific type of bacteria or virus which are causing the disease, they can be even get transmitted by the injection needle also, because if the injection needle has been used for any other patient, and if that needle will be used for for a new patient, automatically if that needle has been used for any sexually transmitted disease affected individual, obviously now from that specific needle the disease will be getting transmitted to the healthy individual. By using the surgical instruments also, these diseases can be transmitted from the infected person, as well as directly from the infected person by the help of sexual intercourse. This specific type of disease can be get transmitted, that is written in the point number 3. If the blood of the infected person will be getting transmitted within the healthy person, on that case also the disease can be get transmitted through the blood. And the last one, that means if any mother is getting infected, obviously if fetus is present within the mother's womb, then the fetus also can be get affected by this kind of sexually transmitted disease, because through the placenta the disease causing agent, it will be getting transmitted from the mother to fetus. Now we will be discussing about their symptoms, whatever the symptoms are commonly found in case of sexually transmitted diseases. There are the various symptoms which are commonly found in case of sexually transmitted diseases. First one, itching in the internal reproductive organs. The fluid will be discharged through the male or female external genitalia. Slight pain will be there inside the reproductive tract or reproductive organ. Swelling can be get created also. Burning sensation can be felt during the time of urination, and abdominal pain will be created in the specifically genital regions. So these are the most common symptoms found in case of these sexually transmitted diseases. Now the point is, this could lead to the complications, such as whatever the complications can be found. The complications are, for these sexually transmitted diseases, pelvic inflammatory diseases can be occurred. Abortion has to be done to reduce this kind of problems of sexually transmitted diseases. Stillbirth can happen. In case of some of the female, ectopic pregnancy also can take place. Ectopic pregnancy means actually during the time of pregnancy, that growing fetus or the growing embryo is present within the female womb or within the uterus, but if the implantation of the blastula has happened to any other area, instead of uterine lining. So on that time, that pregnancy is commonly known as ectopic pregnancy. So that can happen. This kind of STDs are causing infertility also for the female, and this kind of STDs are also causing cancer or in the reproductive tract. So abortion, stillbirth, ectopic pregnancy, infertility, cancer in the reproductive tract, as well as pelvic inflammatory diseases, these are the complications can arise due to these sexually transmitted infections or sexually transmitted diseases, or rather reproductive tract infections, RTIs. Now, simple principles for avoiding STIs. That means if any individual is suffering from the sexually transmitted infections, whatever the simple principles are present to avoid that. First one, avoid sex with the unknown partner or multiple partner. Obviously, if we can avoid the sex with the multiple partner, unknown partner, obviously this kind of problem of STD can be avoided. Use of barrier method during the time of coitus. That means use of condom, femidome, this kind of diaphragm, vault, this kind of tools or devices we need to use during the time of sexual intercourse, so that we can avoid this kind of sexually transmitted diseases. As well as if anybody is suffering from the sexually transmitted diseases, that individual should consult with the doctor for early detection of the STIs. So early detection of the sexually transmitted disease is very much necessary, because if the delay will happen, it will create more problem. Quick treatment if is needed. So if the detection will be done properly, then very quick treatment it has to be started, because if also treatment will be started in a delayed manner, then the disease can be getting spread within the body of the individual human, and afterwards by providing a higher level of treatment also, that specific individual human cannot be saved. So for that reason, quick treatment is also very much needed for this kind of STIs. So I think the sexually transmitted disease, their symptoms, whatever the processes to avoid those STDs, those things I think very much clear to each and everyone. Now we are just moving for the next topic, that is a new topic, that is infertility. We all know infertility is such kind of problem in modern era which is commonly found in case of male as well as female, and infertility is such a problem in most of the cases which is not getting cured properly, because it's non-curable. So for that reason, there are two types of male infertility we will be discussing today. One is the male infertility as well as female infertility, and not only we will be discussing regarding the various problems in male and female, with that what can be the solution, whatever the various strategies or procedures are actually present to reduce this kind of problems, infertility problems, that also or those also we will be discussing in this specific video. So first of all, we are starting with male infertility. Let's see whatever the infertility problems can be found commonly in case of male. First of all, these are the three common problems which are found in case of male regarding infertility. First one is azoospermia. Azoospermia is a medical condition of a man whose semen contains no sperm. That means absence of motile sperm in the semen. That is the first problem which is told here. That is the azoospermia. Azoospermia means absence of motile sperm in the semen, and it is obviously associated with infertility issue. Next one, oligospermia. Oligospermia means if the sperm count in the semen is less. So if the sperm count in male semen, if it is less, then obviously it is characterized by oligospermia. Third point is asthenozoospermia. Asthenozoospermia or asthenospermia is actually the medical terminology which we are using for making us understand that reduced sperm motility. That means in case of any of the male, if it is found that sufficient number of sperms are present within the semen, but the motility of the sperm anyhow it has got reduced. Okay, so that specific individual human, it will be told that for that person he is suffering from asthenozoospermia or asthenospermia. So now the point is asthenospermia can be of two types, complete and incomplete. Complete means that in that specific person 100% of the sperm is showing no motility or no mobility, but in case of incomplete asthenozoospermia, obviously for that person it will be found that 50% of the sperm or more than that will be showing motility, remaining sperm they will not be showing any kind of motility. Okay, so incomplete and complete, these two kinds of asthenozoospermia or asthenospermia can be commonly found in case of male. So azoospermia, oligospermia and asthenospermia, these are the three different types of problems, regarding male infertility. Now we are entering within female infertility problem. What is regarding the female infertility? Female infertility means in case of female also some of the problems can be found. Let's see the problems. In case of female, the infertility problems are irregular menstruation, second, narrowing of uterus, third, improper endometrium lining, fourth, blockage in fallopian tube, fifth, polycystic ovarian syndrome, and sixth, impotency. That means first one, irregular menstruation. Commonly it is found that in case of nowadays, in case of female, it is a common problem that menstrual cycle is getting irregular, and it is getting irregular obviously for our lifestyle, for our food habit and all these things. But irregular menstruation is obviously one kind of infertility, because in case of those female who are having irregular menstruation, that for those specific type of females, conception is also a questionable task. So for that reason, those specific female who are having irregular menstruation problem, obviously they need to consult the doctor so that this problem can be minimized and they can conceive the baby. Uterus lining, or specifically I want to say that canal of uterus, if it is not so much broader, if it is narrower, obviously it can't provide proper passage for the growth of newborn baby or for the growth of the growing embryo. So narrowing of the uterus is also another problem. For that reason, the female, they remain infertile. They can't even carry any of the baby. Improper endometrium, if inside the uterine lining the endometrium is not formed properly, then obviously for this improper endometrial lining, the female will be suffering from infertility problem. Blockage in fallopian tube, that is the fourth problem. In fourth problem, it is told that if suppose it is found that in the fallopian tube of the female there is any kind of the blockage, if the sperm and ovum, they can't come together for fertilization due to presence of that blockage, obviously that newborn baby will not take birth. There is no growth of the fetus takes place. Polycystic ovarian syndrome, in case of some of the female, this is also very common problem nowadays, that it is found that in their ovary multiple cysts are getting produced, and due to formation of multiple cysts, obviously those females are getting incapable of producing the newborn baby. Impotency, it is obviously making any female impotent, rather you can say their ovaries or any of the reproductive organ or reproductive part is getting non-functional, and for that reason these females are totally get incapable of conception, so they can't conceive a new baby. So for that reason, these are the common problems which can be commonly found in case of female regarding infertility. Now we have come to the solution of the infertility. Solution is only, that is the method or the strategies by which the male and female infertility problem can be reduced, and the mechanism in most of the time is fertilization of the male sperm and female ovum out of the body. So assisted reproductive technologies means those specific type of technologies by which we can fertilize male sperm and female ovum out of the male and female body within a artificial environment by providing all kinds of natural requirements. So this specific type of methodology or this kind of technologies are commonly known as assisted reproductive technologies. So within the assisted reproductive technologies, whatever the various technologies we need to study or what we need to discuss, first is the IVFET, that is in vitro fertilization embryo transfer, that is the first method. Gamete intrafallopian transfer, that is the second method. Third, ZIFT, zygote intrafallopian transfer, that is the third method. Fourth, AI, artificial insemination, that is the fourth method. Fifth, ICSI, that is intracytoplasmic sperm injection, that is the fifth method. And sixth, that is IUI, that is intrauterine insemination, that is the sixth method. So these six methods are there under assisted reproductive technologies. So one by one now we will be discussing each and every method. First we have come to the IVFET or in vitro fertilization embryo transfer method. In this method, what is happening? In this method, actually the male sperm and the female ovum will be collected from the respective male and female individual who are actually the couple or who wants baby. So this after collection of the sperm and ovum, those will be washed properly, and after washing, those sperm and ovum will be kept within a petri dish. And after keeping within the petri dish, proper culture media will be provided, proper environment will be provided, temperature, pH and all these things for further fertilization. If the fertilization will be getting over, after that the zygote will be getting formed, and zygote will be getting increased up to 8 to 16 celled stage. So the zygote will be grown out up to 8 to 16 celled stage, that means up to blastula stage, and just the blastula stage arrived, then the blastula will be implanted within the endometrial lining of the female. So that is the embryo transfer. So this is actually the in vitro fertilization, where the fertilization is taking place outside of the female body. That's why it is in vitro fertilization. And up to blastula development, we will be waiting. Just the blastula will get developed. This blastula will be implanted within the endometrial lining of the female. So it will be the embryo transfer. So for that reason, this method is commonly known as IVF-ET or in vitro fertilization embryo transfer. Let's see the procedure. It can be steps of in vitro fertilization is mainly 5 steps. First of all, we need to stimulate the ovary of the female by the FSH and LH injection. Why? Because if the ovary will be getting stimulated, multiple ovum will be getting produced at a time. So after stimulation, oocyte will be retrieved. Oocyte means secondary oocyte, or ovum I am talking about. So the oocyte will be retrieved or removed from the female body. Next, sperm also will be retrieved from the male body, obviously from the male testicles or epididymis. Then the sperm will be washed properly, and within the petri dish the fertilization will be performed. After fertilization, up to blastula stage we need to wait, and that blastula stage will be implanted into the endometrial lining of the female, and that is the embryo transfer. Then entire developmental process of the embryo, it will take place within the female body. So this is the steps of in vitro fertilization what I have discussed. Next, gamete intrafallopian transfer or gift. What is gift? In the gift, sperm and eggs are placed in a fallopian tube to allow fertilization in the natural site. That means isthmus ampullary junction. In the natural site the fertilization takes place. For allowing that, we are performing gift. The woman must have at least one normal open fallopian tube. If the female should have one at least normal open fallopian tube, then only this gift process can be performed. That means the gametes will be collected from the male and female individual respectively. After that, those will be transferred within the fallopian tube, and fallopian tube where specifically in that portion where the fertilization takes place, that is the isthmus ampullary junction. After transferring, obviously the fertilization will be taking place, and then the further developmental process will happen. ZIFT, zygote intrafallopian transfer. What it should be done here? Eggs and sperms will be collected from the respective male and female individual or couples. The collected sperm is combined with the eggs, obviously outside of the body, that is the in vitro fertilization will be performed. After that, the embryo will be transferred, specifically the zygote will be transferred or inserted within the upper portion of the fallopian tube. That means just after the isthmus ampullary junction, the zygote will be transferred, and from the upper portion of the fallopian tube, when that zygote will be coming towards the endometrium portion of the uterus, within that time the blastula has been formed, and that blastula will be further get implanted in the endometrial lining. So this is actually the target of zygote intrafallopian transfer, where we will be producing zygote outside of the female body artificially, and after that the zygote will be getting transferred within the upper portion of the fallopian tube, and from there gradually when the zygote will be coming up to endometrium, within that time the formation of blastula will get completed, and after that it will be get implanted in the inner wall of endometrium. Artificial insemination, what is that? Artificial insemination is the deliberate introduction of the sperm into the female cervix or within the uterine cavity. So this specific process is commonly known as artificial insemination. Now the point is, during the process of artificial insemination, the sperms can be inseminated or deliberately can be introduced within some of the portions of the female reproductive tract. If the sperm will be introduced in the uterine portion of the female reproductive tract, then it is known as intrauterine insemination. Second, if the sperm will be introduced in the cervix region of the female reproductive tract, then it is known as intracervical insemination. Next, if the sperm will be introduced in the fallopian tube directly, then it is known as intratubal insemination. And at last, if the sperm will be introduced in the vaginal location or vaginal canal, then it is known as intravaginal insemination. So now, which specific type of insemination will be performed by the doctor, that is depending on the structure of the female reproductive system entirely. Which of the portions, suppose uterine, cervix, fallopian tube and vagina, out of these 4 portions, which of the portions are not normal, showing abnormality? So on those specific portions, the semen or the sperm will not be transferred. Which portions are showing normality, which portions are properly present, no problems are there regarding those portions, they are only during the time of artificial insemination, the semen or the sperm will be inseminated, not in other portion. Going for the next point. Next point or the next type of assisted reproductive technology is ICSI, that is intracytoplasmic sperm injection. This is the method where directly the sperm will be introduced, or rather the sperm nucleus will be introduced within the cytoplasm of the egg. So here actually it is found that if the sperm is not having motility, on that time the artificially the sperm nucleus, or rather the entire sperm will be introduced within the egg cytoplasm, and it is obviously the more updated method than GIFT and ZIFT and that artificial insemination, because here the sperm nucleus or the sperm is directly introduced within the cytoplasm of the egg, and this egg is obviously, it should be obtained by the process of in vitro fertilization, because during the time of in vitro fertilization, whatever the egg we are collecting, by that we can continue this ICSI process. Here you can see the process of ICSI. Egg will be held by a tool, and within the egg cytoplasm, by the help of injection needle, we are introducing the sperm. So sperm is injected into the cytoplasm of the egg using fine needle, and that is directly performed. So this is ICSI, and there is no failure of fertilization, because fertilization should be happening in this specific process. Intrauterine insemination, what is that? Intrauterine insemination is a such kind of process where the semen or the sperm will be inseminated within the uterus portion specifically, and that is facilitating the fertilization process. The goal of this IUI is to increase the number of the sperm that reach the fallopian tubes and subsequently increase the chance of fertilization. We have already studied about artificial insemination. Under artificial insemination, intrauterine insemination was there. So if we can inseminate the sperm or the semen in the uterus, we will do that, because the main target of this insemination process is that more number of the sperm, if we will inseminate the sperm within the uterus portion, so more number of the sperms, they can reach up to the fallopian tube, and subsequently increases the opportunity or the chances of fertilization. So obviously for obtaining that, intrauterine insemination is the most appropriate method within the artificial insemination than others. So this is overall the assisted reproductive technologies we need to study under infertility, to avoid the infertility obviously. And before that, I have also discussed about the various kinds of male infertility and female infertility problems. I have also discussed about the sexually transmitted diseases, MTP, medical termination of pregnancy, as well as the amniocentesis. So with discussing these all points, this reproductive health chapter is getting finished. So thank you everyone. I think this entire video, whatever I have prepared, you people has understood properly, and obviously I think you have enjoyed this video a lot. Thank you everyone for listening this video. Thanks a lot.